This is TWIS. This Week in Science, episode number 811, recorded on Wednesday, February 10th, 2021. Digging up science bones. Hey everyone, I'm Dr. Kiki, and tonight we will fill your head with limbs, bones, and vampires. But first, thanks to our Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. When you think about it, whatever it is you happen to be thinking about, keep in mind that the thing you are thinking about has been thought about before. If, for example, you are thinking about how to get a rock out of your shoe without having to remove the shoe, chances are somebody has thought about doing this before. It doesn't mean that others have spent time thinking about how to get rocks out of your specific shoes with your specific feet in them, but certainly others have encountered a similar situation, may have even found solutions. So whatever occupies your mind, think about it, then find out what others have thought about it. Taking on ideas, solving problems with sets of solutions is always better than going it alone. Nowhere is that more true than when talking about This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back. Oh, yes. The full team is here to talk about all the science that we loved this last week. And I have stories about fins to limbs, fractals in vision, a little bit of carbon cycling action, and maybe I got bones and brains and things like that. Lots of fun things this week. Justin, what do you have?
I've got the wrong story up. I was going to tell you last week is what I had. Sorry.
No.
What week is this? Okay, what did I bring? Oh, yeah. I've got a big step in Conch, a warning for humans about evolution, Google Translate fails, why marketers are worried, and why marketers are worried about political polarization.
Polarization?
Polarization.
That has nothing to do with carbonated soda. No. That's carbonization. Yes. Blair, what is in the animal corner?
I have stressed animals. I have upside-down dragonflies and baby vampires.
Baby vampires with those little fangs. Oh,
Yeah.
it's going to be so cute. Can't wait.
I want to suck your blood. Okay.
Moving in to the science show. If you have not yet subscribed to This Week in Science, please do. You can find us all places podcasts are found and on YouTube, Facebook, and Twitch. Look for This Week in Science or TWIS Science. We are out there. Our website is twis.org. All right, let's jump into some of that fishy business. Are you ready for fish stories?
Mm-hmm.
I have an evolutionary story for you. It started before the fin-rayed fishes, the teleosts, and the four-limbed mammals that became us. Once upon a time, we shared a finny ancestor. It was kind of finny, four-limbed. It liked to crawl around, and then it lost that crawly ability and was like, I'm going to be swimmy and fishy.
Oh, it went the other way.
Yes. So once upon a time, this ancestor had lots of little bones leading out from the shoulder joint. And those bones were connected with tendons and muscles through ligaments to those bones. And mammals use that to create limbs, front limbs. Humans, now that's our arms and our shoulders that, have issues because they're not the best design, but they're pretty good. They work
I
okay.
think they're amazing. I think they're good.
Shoulders are pretty okay.
How nice for you.
But this ancestor, at one point, fish were like, nah, I don't need those front limbs. I just need a little tiny pectoral fin. And they're like, I'm swimming, just keep swimming, right? And the bones became less segmented, and they lost the musculature. They lost the tendons, the ligaments, all the connectivity. And the bones just shoot straight out from the shoulder joint as the rays. And those are the fins, basically. The bones sticking out, and there are muscles between them.
Don't use
control
two
those
bones.
fins.
Don't use two bones if one is good. Yeah, I get it.
If one is good. So these researchers, just publishing in Cell, the journal Cell, they have done a bunch of different experiments, which are really interesting to kind of get to understand what happened genetically to lead to this differentiation of the finned fishes and the limbed animals. So they took these fish, zebrafish, which are a part of the teleosts, and they applied a mutagen. They mutated the fish, basically, from the wild type, forced their genes through a mutation, and then bred them a couple of generations back into the wild type phenotype again. And what this allowed them to do was look for weird mutants, weird mutations. And they found that there was one individual of the second generation offspring that had a limb instead of a fin. It looked like it was starting to grow limbs. And they went, that's the one. And so then they went back to the genotype, gene sequencing, and did a bunch of knockout and knock-in experiments to play with a couple of genes that they didn't originally think would be involved in the process of differentiation of the limbs. So we know that Hox genes are involved in creating limbs and fingers and toes and all these kinds of things. But in this process, there are a couple of other genes that are involved, VAV2 and WASL-B, and they determined that mutations in those genes that are further downstream and had never previously been thought to be involved in limb differentiation turned on a Hox gene that led to a gain of function, basically taking these fish that have not had limbs for 400 million years and giving them the ability to start differentiating new skeletal bones with musculature and muscle insertions. So creating joints within their fins. So what they're saying is their research is pointing to this latent ability in fish that if something were to arise that would create a mutational event for finned fishes, they could potentially evolve limbs again. They still have the genes that are involved in that. And then adding on to it, what it also says is, hey, there are these genes that we never thought were involved in these arm bones before, but now here they are. What other genes might be involved that we never thought were involved?
I just simply didn't know that all these fish were from something that had crawled around on land first. I didn't even know that. That was that's amazing by itself.
The common ancestor of the tetrapods and the teleosts. Yeah. Huh. Like a swimmy crawly common ancestor. Yeah. Okay. But a swimmy ancestor that had more jointed limbs in the front, more bones and limbs. Yes. Yeah, but it's pretty interesting that you can take something that is, it's separated from us evolutionarily by 400 million years. And hey, you can still give it. It's like the giving chicken teeth experiments that were done like 10 years ago, right?
Mhm.
We can still do these things. Yes. Evolution's crazy, but we're learning more all the time. Tell me a story, Justin.
Silent for some 17,000 years. Discovered in a cave some 90 years ago, then quickly forgotten. In a dusty box in the musty basement of a lusty French Natural History Museum for the past 80 years.
A lusty French history museum.
I was rhyming.
Okay. All right.
But now a large sea snail shell has been rediscovered. But this large sea snail shell, or conch if you will, is no ordinary shell, or conch. If it was an ordinary conch, I would likely not be telling you its story, as conches usually live very uneventful lives. This conch, however, was found in the Pyrenees. Now, if the Pyrenees were islands, this would still seem like an unremarkable story. However, the Pyrenees are mountains, really big ones that separate France and Spain. In these mountains, there is the Marcellus Cave, which is 120 miles from the Atlantic Ocean and at an altitude of about 6,890 feet above sea level. The cave itself is 330 feet deep, and it's got artwork on it, covered in artwork, 17,000-year-old human artwork on the walls. It's very unique, specific sort of ancient artwork. It's depictions of like bison and animals and everything, and that sort of typical trade, but it's all done in these red dots, like finger-sized dots. Like they did this sort of like color by dots to make these. The caves were discovered in the late 1800s. They excavated then the greater area outside the cave, I think in the 1930s. And around the mouth of the cave, they found a large sea snail shell. What is it doing there? Well, oddly, it was decorated with the same red dots that comprise the cave art. It had a few small, sort of unnatural-looking holes at the tip, meaning this was picked up from the ocean 120 miles away, carried for that 120-plus mile trek up a mountain on foot, decorated, modified carefully to have a hole in it, and it can be played.
So did they push air through it, or did somebody put their mouth on this thing?
So somebody put their mouth on it. So they hired a musician.
Really?
So they did do a slight modification because later versions of these have been found, or more recent versions have been found, that have a sort of mouthpiece that gets attached to it. So they think the hole is actually a place. There's two tiny holes that were drilled in, are designed to attach some sort of mouthpiece to it. But oh, there's the sound. So they were able to get three distinct, notes out of it. But you got to picture this thing is also if it's being played in a cave and by a musician who's had a little bit more time with it, they may actually be able to belt something out. And in the acoustics in there, So anyway, and then to know that like, oh, here's this shell. I know what to do with this. I'm going to walk it 120 miles up a mountain, carve some holes in it and make it into a thing, means this has probably been, going on for a while. This is the oldest, dated around 18,000 years. This is the oldest conch wind instrument ever found. And
it's amazing.
Yeah. It is. It is. So the research on this is published in the journal Science Advances. Pretty fun story of another one of these, discovered 90 years ago. 90 years ago it was discovered and quickly ended up in that box in the basement. How is it? We just need to open up all the dusty boxes in these museums.
I know, right?
There's cool stuff in there.
Can we get somebody to hire a bunch of graduate students to go into the boxes, the shelves, the drawers?
I wonder how many of these are because of COVID, though, because you can't really travel. It's a lot harder to do field work. And so you're just like, what's in the drawers?
Yeah.
I don't know, but this has been the kind of a trend for years where somebody finally gets around to cleaning something out or looking into a box, or a PI or somebody in charge says, "Hey, why don't you go look through this area and see if there's anything interesting? I'm sure there might be." And then you find something.
And and it apparently hadn't occurred to the original people, the original researchers who had discovered this, excavated it. They thought it was interesting that it was there, that it had these markings, I guess. But other than that, they hadn't really put a whole lot of thinking to it. Now, since that time, because this would have been in the 20s or whatever when they first found, or 30s when they found this. Since then, we've seen lots of examples of conch being used as instrument. So it may just also be an update in the ability to perceive the other uses of it.
Let's update things more. Blair, tell me a story, please.
I'm stressed.
What? Why?
And so are a lot of mammals in South America. So
why?
This is a story that is definitely, at first you might consider it a bit of a well duh, but I'd like to just explore it for a moment here. So this is looking at small mammals in South America's Atlantic forest, and it's specifically rodents and marsupials. And they wanted to see what kind of stress these animals were under, especially the ones in areas going through deforestation. The hypothesis was that animals in deforested areas would show higher levels of stress than animals in more pristine forests. And that's what they found. This was from the Field Museum in Chicago and Chicago State University. Small mammals, primarily rodents and little marsupials, tend to be more stressed out or show more evidence that there are higher levels of stress hormones in their hair, specifically in smaller forest patches than in larger forest patches. They trapped 106 mammals from areas ranging from 2 to 1,200 hectares. So that's about the size of a city block to 4.63 square miles. So pretty big difference. And the critters that they looked at, they took samples of their fur, and that allowed them to look at more long-term hormone trends. So if you test blood or urine, it's a snapshot. But if you're looking at fur, you're seeing a much longer-term expression of those hormones. So it's more than just, I caught this animal and now it's stressed, or this animal ran from a predator earlier today and now it's stressed. There's long-term stress hormones hanging out in this animal, therefore showing up in their hair. And so they did find that stress hormones were higher in the hairs of these animals from areas that were being deforested. And, again, this is probably a bit of a well duh, but it is something that's important to consider when we're thinking about impacts on species, because there's the direct impact and there's an indirect impact. The direct impact being they have less space. They have to compete more. They have less spaces to hide in. They have less access to mates, and the gene pool is narrowed, all these sorts of things, right? The indirect could be that this is also making them stressed and therefore impacting their fitness in other ways. And I will also just say real quick, not all stress is bad. Short-term stress is good. Stress is an important hormonal response that our body does. And when it's not happening, there's actually problems. And so the issue is chronic, long-term stress that doesn't abate. And so that's really what they're looking at here. I also just think about my own personal stress levels, having less space over the last year, and thought that was kind of an interesting parallel.
Would you say that? So, what would be an example of chronic lack of stress? Is that what, like, a panda is?
Sure. Yeah, it's more like for example, if I just gave my dog her food in her bowl and her treats in her bowl, and she got everything she ever wanted because I just handed it to her every day, that would be lack of stress. Putting food in a feeder or a toy, making her train for her food, making her perform for food in some way or another is actually a stressor, because she's going like, I want the treat. How do I get it? What do I do? So that is an example of a little bit of stress injected in to help with the mental, physical, just systemic health of a being.
Okay. Yeah.
Mm-hmm.
I want the food. Give me the food. I have stress every day. What am I going to eat? How am I going to cook it?
It's
Who's going to
like
order food for me? No. It's my stress. All right, moving from stress, let's talk about fractals, man. Fractal vision. So fractals, we may be familiar with because of Benoit Mandelbrot and the Mandelbrot set and the popularization of the idea of fractals as making up everything. Fractals are infinitely complex patterns that repeat themselves infinitely. So at the smallest scales, you still find the pattern that is representative of that fractal. And some Japanese AI researchers were thinking about the inherent bias that we see very often in the training of artificial intelligence. And so currently, if we are going to be training an AI, what we do is we set it to a visual set of images from Google, for instance, a picture set, something that has been created. Maybe it's landscapes, maybe it's people, maybe it's cats, but there is a training picture set that helps the artificial intelligence figure out what lines are that differentiate between the subject of an image and the background, or other aspects of vision. What makes vision turn into comprehension of a scene and its contents. So these researchers looked at the idea of using fractals to train computers instead of potentially biased image sets, because the images that we give to these computers potentially can be biased because of who put the image set together, because of the images that are included in it. And to create something completely unbiased, they thought, hey, let's try this out. So they created a fractal training set and pre-trained their artificial intelligence, vision intelligence, to see things. And they said in most accounts, it worked almost as well as using real images, which is pretty cool. And it kind of gets to the point
that it found fractals just as confusing as
we do. Yes,
as we do, but found everything that we look at just as confusing as a fractal.
Yeah, I think that's probably more it. Everything that we look at is as confusing as a fractal. But the idea is that there are these infinitely naturally repeating patterns in nature. Tree limbs are fractals. Clouds are fractals. Lightning is a fractal pattern. Just about broccoli is a fractal.
What?
Yes. When you have
broccoli is broccoli all the way down?
Broccoli is broccoli all the way down. Exactly. Wow. But you find evidence of fractals throughout nature. And so it's not perfect for sure. It's not a perfect teaching set, but they did find instances where it was as good, if not better, than the picture sets that have been created for AI training in the past and are like the standard sets. The reason I brought this story is that I think the concept is very interesting because they're trying to solve the problem of the unintended bias in artificial intelligence vision systems, in the visual learning systems. What do they identify? Are they identifying it because the training set was biased because humans made it? Or are they identifying it because they've learned rules about vision and what makes a thing a thing? And so I think it's a very interesting way to address this problem, and we'll see where it goes in the future. It's fractals all the way down, man.
Yeah. Okay.
Yeah.
It still feels like there was this math class once. If you weren't a science major or accounting, I suppose, you could do a math class if you're doing liberal arts, I guess. You could do a math class, which was just going in and watching fractals. It was like movie fractals. And then you would discuss, and that's all you did because they just were like, you're never going to do math. We get it, but it's a requirement. So here's something that sounds like math. That's a movie you're going to go watch. That was at a college. That was a college level course, was watching fractals.
Non-major courses, some of the best.
But it was one just great. You can't go further with this.
It
This
gets you
doesn't
into
build.
it. You're like, oh, it's a taste.
Isn't,
That's
this
right.
isn't one of the steps in building towards a higher math, but this will qualify for you to not do math again.
All right. Take me on
Take it
to another
twice.
story, Justin. What do you have?
This
some
is
some evolution?
Yeah, this is actually a warning for those from those who study human evolution. They're saying, we are far from done working on this. We're not done. This is actually
What,
quite
this? You just mean this? We're not done?
no,
The
we're not
human
done.
body or studying human evolution?
The understanding it. Yeah. So this is actually a pretty heavy hitting group. This is experts from the Natural History Museum, Francis Crick Institute, and the Max Planck Institute for the Science of Human History. They've gotten together, teamed up to untangle all of these crazy data points that we're getting about Homo sapien origins and ancestry. In the new paper published in Nature, they review our current understanding of how modern human ancestry around the globe can be traced going backwards, which ancestors it passes through during that journey back, with some specificity here and there. This is the part that caught my eye. The co-author, researcher in the Natural History Museum, Professor Chris Stringer, said, some of our ancestors will have lived in groups or populations that can be identified in the fossil record, whereas very little will be known about others. Over the next decade, that's the one that hasn't happened yet, growing recognition of our complex origins should expand the geographic focus of paleoanthropological fieldwork to regions previously considered peripheral to our evolution, such as Central and West Africa, the Indian subcontinent, and Southeast Asia. So that was the big part. They're finally kind of acknowledging that, yes, the places in Eastern Africa where it's been very easy to find fossils because they're great at keeping fossils might not be the whole story of human evolution. It's just where we've gotten the most data from, right? But yeah, they're arguing that there's no specific point in time can currently be identified with modern human ancestry where modern humans were confined to some limited birthplace. And that the known patterns of the first appearance of anatomical or behavioral traits that are often used to define Homo sapiens actually sort of fit a range of evolutionary histories. We got cousins everywhere. If we're even us, we might even be one of the cousins. We might have gotten it wrong. What we think of our current origin might actually still be cousins to what it becomes.
Sounds like you're talking about a braided stream.
It very much is the braided stream. But what they're also sort of highlighting here is there's been enough mystery. One of them is that little pinky bone from a Denisovan found in Altai Mountains, which is a Siberian cave, right? There's a Neanderthal there that was also found. That Neanderthal, from a different age, that Neanderthal apparently had modern human ancestry. 200,000-year-old fossil in the mountains of Siberia. So the out of Africa thing is still part of it, but the when and what waves and what interminglings, and yeah, they're just basically saying that whole idea that there was a this, then a this, then a that, then a this, then a that, even after the braided stream, even after a lot of that braided stream took place, still not done. Still going to be more. Anyway, I thought it was a fun warning that the next decade is going to... Also, they say the success of direct genetic analysis so far highlights the importance for a wider ancient genetic record. They want to continue to improve the ancient DNA retrieval, biomolecular screening, fragmentary fossils, also to do wider searches in areas that they have not been looking at in the past, previously. They're always looking in the past. where they haven't looked previously, using sedimentary, which is that thing when they sort of dig into, which I would love to, that would be one of the wonderful finds we might be able to get out of the caves in Mexico, those 30,000-year-old caves that we're just discovering, where they go down into the sedimentary dirt and actually sort of filter that through a genetic sequencer. A lot of noise, but they've gotten really good at getting signals out of that, whether something is human or Neanderthal, or they can sort of date whether or not that was different layers of sediment actually have human DNA somewhere in them.
It's just sitting there, old spit, waiting to be identified.
So if you want to study this stuff and you're like, all the good bones have already been found, they're saying
dig some dirt.
They're saying no. This field work is just at its infancy. This is the launching point.
Yeah, we humans are all over the planet now. We didn't used to be, but we haven't at this point in time gotten to the point where we're digging all the places where we used to be. So that's really what we need. More digging. We just need to dig in the dirt some more, everybody.
And when you
go back to our toddlerhood.
Used to be, we keep having to go further and further back in time to say we weren't there. And
true.
Also, then we also have to very clearly define who we are. Homo floresiensis, hundreds of thousands of years ago, in places that we thought humans got to much more recently.
Yeah, I love it. I am looking forward to the future of human evolution fossil finds. This is going to be fun, everybody. All right, I have a story about allergies first. Yes, because I don't know about you, but I personally, for the last few years, have been experiencing more severe allergies.
I thought you were going to talk about him. I thought you were going to say more severe toenails.
What? Allergies.
Allergies. Is that what we're talking about? Okay.
Allergies.
Oh, I'm reading in the chat room. That's why. I'm reading and half hearing you and half reading the...
I said nothing about toenails.
No, the tenor said toenails are forever. Sorry, I was reading that while you were talking.
So are teeth.
I'll stop reading.
Teeth and toenails, yes. But I'm talking about allergies. And I feel as though my allergies have gotten worse in recent years. I am aging, so my aging immune system should be getting less reactive. I was hoping that with age, I would find fewer allergies in my life, but that is not the case. And I've been wondering, I'm like, man, they seem like they start earlier, they last longer, there's more going on, and they're just bad. What's going on? I spend half the year with allergies, at least. Well, I am vindicated by a new study in the Proceedings of the National Academy of Sciences that has, yes, found confirmation that our allergies are worse because the allergy season is longer. There's more pollen being produced by trees. Their analysis of pollen count stations in the United States and Canada between 1990 to 2018 finds that the average pollen season starts about 20 days earlier than it used to, runs 10 days longer on average, and pumps out 21% more pollen. Thank you for that sentence, Gizmodo.
20%.
Yeah, 20% more pollen. It's an increase.
Yikes.
And there is a difference in which species, locations, while they're talking about all of the United States and Canada, the Midwest and areas like Texas are among those more strongly influenced by this change. But the biggest increase has happened in the more recent decade. So in the decade from 1990 to about 2010, there wasn't as big an increase in pollen or a length of growing season as there was between 2000 to 2018. And they predict that with climate change, we will see even worse allergies. It's not ending anytime soon. So I do hope that we come up with some better allergy medications. Okay, science, get on it. That's what we're going to need. But yes, The allergies. They are on us. And if you feel like they're bad, they are. They're getting worse. Thank you, climate change. And then my last intro study, I wanted to talk a bit about a new carbon cycle that has been discovered in the oceans. It's not just
new one.
Yes, it's in addition to the carbon cycle that already goes through the oceans, but some researchers who were interested in the oils that are in the ocean, they started looking at hydrocarbons, knowing that there are oil seeps that are natural. We have human-induced oil spills. There's all sorts of oil in the ocean. And yet a few years back, these researchers discovered that cyanobacteria in the ocean release a hydrocarbon called pentadecane, NC15. That's 15 carbons, pentadecane. So they started looking at these little cyanobacteria and the pentadecane that they release and looking at the cycle that they undergo, trying to figure out, all right, well, we know that there are some bacteria that like to eat petroleum. Are they related to the bacteria that eat this naturally released pentadecane? And so they started doing a whole bunch of work to figure out, to quantify how much pentadecane is in the oceans. And they determined that there's actually a very high quantity of this hydrocarbon in the ocean, that this is a big amount. The hydrocarbons are spread across 40% of the Earth's surface. And yes, there are other bacteria that eat them up. They estimate from their study that the pentadecane is about 2 million metric tons in the water at any given time. There would be more, but because there are so many microbes that depend on this pentadecane that are gobbling it up and creating carbon dioxide for the cyanobacteria to use again in their photosynthetic energy producing cycle, it's a new hydrocarbon cycle. So what they have put together is that this is a biological cycle wherein the cyanobacteria harvest light, produce the hydrocarbon. There's archaebacteria and bacteria that consume the pentadecane, produce CO2, and then that goes right back in. And so there's a constant cycle in the oceans. And they did discover that these bacteria and archaebacteria, they don't like eating petroleum. So it's completely different groups of organisms. So that idea about using those organisms to clean up our oil spills, that's not going to pan out so well. But whole new hydrocarbon cycle that we had no idea was there because we spill things in the ocean all the time. So when ships are going out to sample oil in the ocean, they're not taking care of where their oil or their gas from their motor is leaking. They're not paying attention to hydrocarbons that are being released from paint on the surface of the boat. They're not paying attention to all sorts of factors. And so this study, they controlled for all of that. They made sure they drove their boats in at a very specific angle to the current and the wind. They made sure that the engine was pointed downstream away from the area that they were sampling. They didn't allow any creation or use of hydrocarbons on the surface of the boat while they were doing the sampling so that they could make sure that what they were getting was actually biological in nature. But then, of course, they did gas chromatography to determine that it was biological in nature. But they jumped through all sorts of hoops to do this work and discovered something new about our oceans. Yeah, very cool. New things for the ocean, old things for the sea. I'm singing songs now. This is This Week in Science. Did you just tune in? Yeah, this is This Week in Science. We are talking about science. Thank you so much for joining us. Hey, you want a Zazzle shirt or a mug or something like that with a TWIS logo on it or a picture, one of Blair's pieces of art? Yes. Head over to twis.org, click on our Zazzle link, and you can help support TWIS and also be very stylish and enjoy your TWIS gear. Faux show. Hey.
Yeah.
Hey, hey. I feel like I'm going to be a meerkat going, hey. Or,
Huh?
Oh,
Hey.
wait, what
Hey.
time is it?
Yeah, that's what I was going to say. It's time for Blair's Animal Corner.
With Blair.
She loves our creature, great and small. 5-peg, millipede, no pet at all. You wanna hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
I have some sleepy dragonflies that I want to drop upside down.
What?
Yeah. This is a study
Aye.
from Imperial College London. They wanted to know how insects that fly are able to keep stable in the air. So if you drop a cat, for example, they actually will rotate around a head to tail axis to get back onto their feet. That's the thing that cats are famous for, right? Is always landing on their feet. So they kind of do a sideways roll, like when you ask your dog to roll over. But a lot of insects and aerial animals have been seen doing something more like a head over toe barrel roll. And so researchers wanted to see how dragonflies make sure they are oriented properly if they are in the wrong position, how they orient themselves mechanically, what's happening there, and if it is a conscious or passive effort. And of course, this is just to understand more about the natural world, but also, of course, to make better drones. I feel like that's what it always is about when we're studying animals, especially flying animals. We're trying to make better drones, which honestly,
Of course we are. Yeah.
So
Sleepy dragonfly drones. What?
yes. So the way that they were able to test this and see exactly how dragonflies can orient themselves and right themselves midair is they took 20 common darter dragonflies and they put them, I like to imagine they were like little vests, but they had tiny magnets in them and they were covered in motion tracking dots like you use for, like Andy Serkis would use to be Smeagol in The Lord of the Rings, right? So it's like the motion capture suits kind of. So then they would use those magnets to attach the dragonfly to a magnetic platform, either right side up or upside down. And then they would vary it with tilt on different subsequent attempts. And then they would release the magnet into a free fall so that the dragonfly had to then right themselves. Then they used the motion tracking dots to create 3D models of how the dragonfly moved. And then they captured high-speed cameras of 3D images so that then they could reconstruct all of it afterwards and analyze and figure out exactly what was happening. So the conscious dragonflies, when they were dropped from being upside down, they somersaulted backwards to regain their right side up position. Dragonflies that were unconscious, they anesthetized them. They also completed the somersault, but more slowly. So first of all, that means there is something going on that they don't have to be awake for it to work, right? So then you're wondering, is it just an inherent piece of their body structure that allows them to right themselves, almost like a punching bag that's weighted on the bottom, right? But the way that they tested that, gotta love science, you gotta figure out a way to test this, right? They had to use dead dragonflies and then fit them with the magnets and drop them and see what happened. And they did not maneuver at all when they were dead. Unless...
You know what? Same thing was true when they did that study with cats.
Yeah, see, exactly. But if their wings were posed ahead of time into a position similar to something they saw living dragonflies posed as, then they were able to right themselves, but there also was a little bit of movement around the vertical axis. So they were a little wonky, but if they posed them correctly ahead of time, then they kind of righted themselves. And then, of course, they also, for good measure, you got to control your variables here. They did, in fact, try a dead wingless dragonfly, and it just fell.
What?
So
That's
Who would
surprising.
imagine? Because
you
dragonflies
gotta check,
dead.
guys.
I mean, you got to check everything, but I mean, did they think dead wingless dragonflies were magic?
No,
Wait a
I
sec.
think they just had to
Wait.
control their variable.
Hang on now. Did they do just the wings with no dragonfly body? Because that's the real test. And until you've done that, all of their results are invalid.
It's like a paper airplane, right? But so their conclusion, of course, the reason for this story was that their maneuvering relies on muscle tone and wing posture, and that it is a passive stability. So the beauty of passive stability, it's the reason that an airplane, if their engine stopped, wouldn't just fall out of the sky. They're designed specifically so that they can coast for a very, very long time, fairly steadily with no engines. So passive stability is the same thing that's happening here. And that lowers the effort of flight, the kind of the energy requirements. And it also is likely to have influenced how their overall body shape evolved. And they also use that passive stability most likely to create an advantage, as in general, that's less energy, and they're more able to recover from inconvenient events. But what I think is also really cool is their next plan here, this research team, is to investigate how passive stability has an impact on their active vision. Because if you're not physically moving your body, then it potentially would be harder or take more effort to track your movement with your vision, right? So there's a certain amount of coordination that's tied with us. If I'm walking in a particular direction and I make the conscious decision to turn, I will also turn my vision in that direction, right? But if you're being turned, you kind of have to play catch up the whole time. So there is a question of how their vision and their ability to continually track a vision is affected by it being a passive stability and not active. And then also their strategies in prey interception and avoiding obstacles if they're using passive stability. So at what point do you kick in manual from autopilot to avoid an obstacle?
Right before it's too late.
Right. Right. And so what's happening in their brain, if you want to call it a brain, really is just like a little ball of nerves.
That's what a brain is. A ball of nerves.
Yes, it is a brain, but in terms of it being a brain, like we consider a brain, it doesn't have the same regionality or anything else that we think about with brains with insects. It really is just a nerve ball. But ultimately, yes, you can call it a brain. But yes, so this is very helpful when we're studying any animal that flies, but also, of course, this could push towards better drones. If you have drones that have passive stability and are able to right themselves just based on their structure without exerting energy, that would be very helpful.
Yeah, without having any external control, without having to use too much energy also, because with any drone, energy conservation is going to be important.
Or is this like as the battery fails and you've got it 300 feet in the air, you want it to find a way to sail back down?
Totally.
That's the one. That's the one. Your wingless dead dragonfly drone sailed to me nicely.
So on from dragonflies to bats, vampire bats. Here's your baby vampires. This is a study that comes from the Smithsonian Tropical Research Institute. And it was a study, it was actually part of a larger study looking at camera footage of vampire bats in a captive scenario. They wanted to see just how bat relationships are built and how they treat each other. And it was part of this larger situation. And then this kind of interesting thing happened, sad, but very interesting. There was a young vampire bat pup that was orphaned throughout the course of this study. Now, this means there was camera footage and social dynamic data for days leading up to the event and then thereafter. And so it gave a really interesting opportunity to give an entirely anecdotal instance, a case study kind of how an orphaned vampire bat is then adopted by another female. So this, as I said, it was about 100 days of surveillance camera footage from the Smithsonian Tropical Research Institute. And what happened was, after this mother bat died, another female stepped in to adopt the baby. And in terms of bat terms, obviously there's nothing legal going on. But what that means is that this female spent a long time, a much higher than normal amount of time for just a random female and baby, both grooming and feeding this baby. Now, let me explain here what this means. This other bat started lactating and ended up feeding this baby milk. She didn't have any babies, but somehow this new bat was able to nurse this orphaned bat, which is crazy. So let me tell you the whole thing here.
Hormones.
Yes, hormones. Absolutely. It's always hormones. It's always hormones. So it was a 19-day-old pup. Her mother, Lilith, unexpectedly died, and then she was adopted by another female named BD. And shortly before Lilith died, the pup was spending time climbing onto who would later become her adopted mother, BD. And this most likely initiated the cascade of neuroendocrine mechanisms that caused her to start lactating. She was just physically a lot of contact with this other bat. But as I said, she wasn't pregnant. She didn't have her own babies. But she was already lactating the day that Lilith died. So it's not even that there was like a vacuum of hormones and she filled it. There was some sort of, this other bat is dying, and everyone around them could tell, or that there was something else going on where the baby was rejecting the mother, or the mother was rejecting the baby, and so the baby was getting pushed to this other. This is the big question, right? Is like, what caused this to happen ahead of time? But she was already lactating the day that the original mom, Lilith, died. And after Lilith's death, in addition to nursing, BD was grooming and sharing food with the pup more than any other female in the colony. Now, when all this happened, that's when they decided, like, let's go back and look at the footage from before this happened. And so it turned out that BD and Lilith had been primary grooming partners already.
Aww.
But BD was also Lilith's top food donor. So remember, vampire bats have this very cool thing where if somebody doesn't get enough food, somebody else will vomit blood into the mouth of a friend.
It's a donation.
Oh, nice.
It's helping.
Yes. Yeah. And you can listen back. I'm sure you could search for it on TWIS.org. But we did a really cool story about the dynamics of that, and that there's an anticipation that if I'm helping you today when you're in trouble, then you'll help me later when I am in trouble. There's a reciprocity involved. And if somebody does not reciprocate, then they are less likely to get future blood meals. So there's a very cool thing there, just as a side note. But
yeah,
so it's a complicated social thing is when they share these blood meals. So BD was giving Lilith a lot of donated blood meals, but Lilith did not share very much with BD, which is interesting. They don't really know what that means or anything, but interesting little anecdote. But yeah, in the end, BD was helping the pup way more than any other female, even before Lilith died, and then thereafter. So is this motivated by being in captivity? Is this a one-off situation? Does this happen all the time? We don't know. But here's a case study to look at, to kind of take out into the wild and maybe into other captive groups and see what's going on.
I remember a story that you brought a while back also talking about the small kind of subgroups within vampire bats. Maybe it was flamingos. Maybe I'm getting animal stories confused. But there, I remember a story. We've talked about the reciprocity before and how there's this trust between the bats and they build relationships based on that. They're not going to know every single bat in their, 1,000, 10,000 bat colony, depending on how big it is. They'll have a subset of individuals who are the individuals that they spend the most time with, that they are really sharing their lives with. And I would imagine that BD, as the mom bat declined, became less able to feed the pup, and the other bat just slowly started stepping up, which is amazing.
Yeah. So one of the reasons that vampire bats are such an interesting case for this is that compared to other bats, they have incredibly high investment in their offspring. So the grooming, the nursing, the donation of blood meals as they learn how to go get their own, all this kind of stuff. It's such a high investment that this might be kind of a perfect storm for that. That if a baby has lived long enough that they're, kind of well on their way to adulthood and maturity, it would be pretty devastating to a group to have lost all of the effort that has gone in, because it's also not a one-on-one. So you don't want to lose your investment when you have so much going into that baby. It makes nothing but sense to step up and take care of that baby if their mother is lost. But it is interesting that they had created their own little social group already.
It's wonderful. Yeah. It speaks to the social nature of the bats and and that willingness to do that.
So
biology,
I want to hear more about.
fascinating.
I want to hear more about this story. Like, what happened? What happened? Like, when the baby grew up and left? Like, did BD find somebody else to hang out with all the time, or was BD like all alone? And like, what happened?
Well,
I want to keep watching.
This is a 3-month study where they pulled these animals from 3 different sites across Panama. All of the bats were unrelated. They had never met before. And so I am not sure what happened to them.
So they're not even like, interesting.
Yeah. So it's possible they were released into the wild. It's possible the study is ongoing. I do not know. Good question.
I'm looking at a quote at the end. Did you read this?
Oh, yes. Yes. You want to read it?
As a new parent myself, I've come to realize the utter power of baby cuteness. This is one of the researchers who said this. I feel that my brain has been completely rewired. Most of us can understand the strong desire to adopt and care for a cute puppy or kitten, or to take on the ultimate responsibility of adopting a child. Regardless of why these traits exist, it is inherently fascinating to consider the neuroendocrine mechanisms that underlie them, the stimuli that trigger them, how they differ across species or individuals, and how these traits might even be pre-adaptations for other forms of cooperation. Yeah.
There you go. Even baby vampire bats. Cute.
Cute little fangs. I'm going to suck the blood. Yes. Beep, 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 beep. Beep, beep, beep. I've got little fangs. So many little fangs.
I could just go on and on and on, but I won't, because it's the end of the Animal Corner. And I just want to say, this is This Week in Science. If you're just joining us, or if you're watching it for a while and you were like, what is this program? This is This Week in Science. We speak about science and the current news in science every week. Thank you for joining us. Thank you for listening to TWIS. And if you enjoy TWIS, consider supporting us on Patreon or PayPal. If you head over to twis.org, you can click on the Support Us on Patreon link, and that takes you over to our Patreon community page where you can choose your level of support. $10 and up a month, and we will thank you by name at the end of the show. And we do have annual subscriptions, so you can pay it all at once a year, if that's what you prefer. Over on the website, twis.org, there are also links to PayPal, where if you prefer PayPal, you can pay that way. But your support really does keep this show going and allow us to do what we do every week, to replace our equipment, to do outreach, to try and reach new audiences, and try and talk sense and science. Have some laughs. Be serious sometimes. But it really does help us to bring our perspective to this world that is currently very full of misinformation. We try to be that credible, sane perspective for you and for others. And you can help us bring that perspective to many more people around the world. Thank you for your support. We really cannot do this without you. All right, Justin, do you want to tell a story about Google? What's going on?
This is a study that finds academic research engines, or search engines, especially Google
Googer?
Googer.
Well, I messed up research engines. So the problem is it's search engines for research. Specifically, Google has one. It's called Google Scholar. There are some folks that wanted to study how the algorithm ranks things, and you're not allowed to know. Like, Google Scholar won't tell anyone how they rank these things. Now, it's obviously has something to do with the quality of the work based on how many times it's been cited or how long it's been out there without being refuted. I guess. Nobody really knows. So the search engine optimization that is being applied to these things seems to have a preferred format. And things that are not submitted in this format were found by the study, 90% of them to be almost completely invisible, regardless of the quality of the work or how many times it had been cited. What is that format? Turns out the format that is preferred is English.
That's no good.
Papers not in English. Studies not in English.
Don't get shared.
Don't get shared. And this is through a multilingual search. Like, it's not a English-only search that this was done under. So, apparently you can do both. I don't know. Yeah, a study found the research paper is not in the English format. It has a 90% chance of being completely invisible in multilingual searches. Study authors are members of the Department of Communication at UPF Barcelona. To implement this optimization, we need to further our understanding of Google Scholar's relevance ranking algorithm so that, based on this knowledge, we can highlight or improve those characteristics that academic documents already present and which are taken into account by the algorithm, says Riviera, first author of the study. So, again, Google's not giving it up. All Google data, by the way, is the wet dream of communication majors. All people who study communication want to get inside of the data stream of Google and see what's really going on in the world. And they should definitely.
They want to know the algorithms.
Yeah, but what was interesting about this too is for the study, the authors applied an inverse engineering research methodology. Blah blah blah. They have all these methodologies based on statistics and correlation coefficient nonsense. Communication people have too many tools. But basically what they found at the end of it was that even when the searches were in the multilingual format, for terms that were not at all language specific, the name of a molecule, which is not said differently in any other language, a copyrighted product, which is, the same in any language that you would put it, all of these sorts of things. It still would if you were seeing a language a paper that was in French or German or Spanish with 1,000 sightings, it might be put way down the list with an English one that had 10.
Right.
So there's something aside from the specific search words that could be different in a language. I get if you're putting in English words for your search, you might expect something like that to happen. But if you're specifically avoiding that and it's still happening, then you might have a problem.
That's frustrating to me because I feel like the internet's gotten so good at translating things. Like
Yeah.
Google Translate is pretty good.
It's amazing.
Google's job of translating entire web pages is really good. Social media platforms will now auto translate posts from people that post things in different languages, and it's pretty accurate. So it's pretty frustrating that doesn't apply to this.
Yeah, so they published in Future Internet. They're saying the chances of being ranked in multilingual Google Scholar search increases remarkably if the researchers opt for publication in English, which means presumably this paper was published in English. I don't know why they would do anything else after learning that.
Yeah, I mean, this gets at a very, central problem, I guess, in science publishing in the first place, which is that the majority of papers are written in English. If authors do want to try and get broad international pickup citations of their work, publishing in English in an English language journal is going to usually get them much more coverage than otherwise. However, there are national language-based journals for all disciplines, and I know this is very interesting when it comes down to it, because we've talked for so long about English being like the language that everyone speaks around the world. It's the one that so why not? But the prioritization even of a paper with a higher impact factor, published in a journal with a higher impact factor, with more citations, being less prominent on these Google searches just because they're a different language. It strikes at something like bias.
Yeah, well, that's exactly what I was thinking too, right? Is that like, how many different ways have we said that if there is any homogenous nature of a group of people doing research, there is bias, there is a slant in the results, whether you realize it or not, even if it's just people studying bird calls and finally women going, female birds sing too.
Yeah.
Well, yeah. Current facial recognition will identify a white male faster than anybody else.
Mm-hmm.
I think that's gonna at some point change to a Chinese male, because they're going to be utilizing the technology more. But there's still always this inherent bias of whoever's sort of creating the thing,
right,
uses it in their homogenous group first, and that's how it sort of gets perfected. Yep. So I kind of get like why this is a big problem. The way the communication folks sort of looked at it, because they're at Barcelona, they're looking at in terms of, hey, this is not giving enough exposure to people who are writing in their own languages, and you're not being able to see research in your native language that might be relevant to what you're doing. But it's just such a massive drain also for all research if you're just trying to find anything related to your work, and it's 90% completely invisible, like falling below the even register of being able to track it in Google Scholar. That's a big problem.
There's a huge disparity. Yeah.
Mm-hmm.
Yeah. So speaking of disparities, what about political polarization?
Oh, yeah, this is a fun story, especially after...
Let's go straight from one to another. I like it.
Yeah.
I see where your mind is this week.
It might be. Political polarization is having far-reaching impacts on American life, according to the University of Wyoming and 5 other universities across the country who are... this is papers that they've done, as appears in the Journal of Public Policy and Marketing of the American Marketing Association. So this is marketers and business people talking about how polarization might be a bad thing. And that's how you know it's really gotten bad.
Yeah.
When the people whose job it is to
sell
pigeonhole,
strong reaction. Yeah.
To pigeonhole you into a certain specific person that will have a strong reaction. And they're like, yeah, this is actually not good. They go on. I'm not even going to go through all this. They're pointing out some stuff. that is sort of interesting, I guess, that the political polarization is real, that different sides of the political spectrum are now overlapping less and less on what were maybe shared issues or shared concerns. I think the environment might be one that was a pretty well shared concern at one point. Whatever. The research has shown that political identities, Republican, Democrat, liberal, conservative, help determine people's behavior, attitude, and perceptions. Those identities can be reinforced by people selecting social groups with shared belief systems, consumption of media that only align with those beliefs. Even creation of group-specific shared reality. That sounds familiar, doesn't it?
Oh, yes, it does. Yes.
Group-specific shared reality. This also makes it more difficult for elected officials to effectively govern. That's due to a number of factors, including a lack of trust in scientists and policymakers, as well as too much misinformation in the world. Way too much. So, yeah, one of the things they also point out, it's bad for some businesses. They pointed out to Goya Beans. My Pillow? Yeah, My Pillow guy got involved in politics. That's all he did. And now there's a backlash. Home Depot, the owner of Home Depot, there was a backlash, even though he doesn't control the company of Home Depot. Chick-fil-A got boycotted for some stuff. Seems to be all for conservative causes that they've listed here.
The Hobby Lobby at one point, yes.
Yeah.
Many.
Ultimately, according to researchers, consumer welfare suffers because of political polarization. So those who market to consumers are like, hey, this is not good for you. Try something else. One of the things they point out, those finances, people will take less money to work for a company that they politically idealize, that they have a political symbiosis with or whatever, that they believe reflects their political views.
Yeah, it fits their identity. Absolutely.
Fits their identity. They will take lower wages and also even to work with politically like-minded people. They will select over higher paying jobs. And of course, this can happen in any direction politically. It also says people have the potential to prevent others from becoming friends who have different political views, which they think can lead to a less intellectually diverse community. Health-wise, your political views can affect health, you think? We've seen pretty good examples of people having political, idealized identity, politic views of handling a pandemic. This is the one I thought that was the most interesting, though. Societal interests. For instance, this is quoting, for instance, beliefs relating to global warming, affirmative action, wealth inequality, and gun control often tend to reflect individuals' political affiliations rather than a deliberate processing of relevant information that results in evidence-based decision-making. In addition, the broader negative impacts of these policy areas on society as a whole have the potential to harm individual mental and physical health over the long term. Yeah, affirmative action is really bad for people's mental health as a long-term policy. So even in their attempt to describe the ill effects of politicization in society, they have absolutely biased and politicized their own paper.
A bit. I mean, there is, if you are within an individual within a political sphere of influence that is diametrically opposed to your personal beliefs, your political leaning, then that is going to add stress to your interactions, to add stress to the way you think about things. So it can influence mental health and general health. I absolutely see how this could be possible for either side. So that there are these larger scale aspects. It's not just people talking with people. It is this larger scale influence on individuals. Individuals influence the whole, and the whole influences individuals.
That makes sense.
Yeah.
Yeah,
It's kind of the way I would see it.
that is the big difference too, is that our political and societal ideals are on display for everyone.
Mhm.
And
Now.
I think about the generation prior to the internet and social media, and you could have a friend that was a 180 from yourself in terms of political views, and that I think the different ends of the different sides of the aisle were closer together than they are now, of course, which
Totally.
is part of it, but also you could choose to not talk about that, and you also didn't have their views up in your face every time you logged into Facebook or something like that. And so it would be much easier to kind of have a dissonance, I think, within a relationship.
Yeah, I want to know how marketing can fix this, how we can use social psychology and marketing psychology, and how we can use it to turn it around and get what used to be the Apple versus PC advertisements that were very
Yeah.
Was it,
Pepsi versus Coke.
Pepsi, these societal schisms, right? Why don't we start bringing brands together, showing how you can have integration and you can have cooperation? And I mean, I've seen those I-5
Pepsi
corridor
and Coke commercial?
No. I'm just thinking about, I don't know, they're like Pizza Hut and Taco Bell places that are like together.
But they're not competitors. That's the thing. They're not real competitors.
All right, then I want to see vending machines that sell Coke and Pepsi.
So that is actually, I think that might be some of what they say. They suggest some measures to limit the effects of polarization, including reducing the spread of misinformation, which we've talked about as well. Use messaging techniques that try to bridge different values of liberals and conservatives.
Yeah.
So, maybe have a commercial with like a box wine and then like, a Bogle or something.
Box wine and a 6-pack. I don't know.
Limiting the length of political campaigns. That sounds pretty cool. But at the same time, they say polarization has been shown to increase voting and political participation, which is actually, I think, is the problem. I think those part of what happened in those oldie days when you didn't have as many friends talking about politics all the time is because nobody knew about it or cared about it. And I think
people were complacent also, and
complacent, but comparatively, now people are talking politics without the policy involved at all.
It's sports teams and not endpoints. Yeah,
tribe. Yeah, I liked it better when they weren't participating, whoever they are, left and right. Like,
well, I think it would be amazing for people to be talking about local policies. What about the reservoir up the hill? What about the salmon runs in the
your local news doesn't cover it,
right? But that is another issue, and that is media and all. Yeah, okay, culture. We got to get back to the science, but yeah. But yeah, I agree. Interesting study that you have brought there. Let me talk about bones. I'm going to talk about happy bones. I want to have happy bones as I get older. I imagine you do too. Many people end up with osteoarthritis, which is a deteriorative condition in which a lot of people think that it has to do with bone injury and then cartilage getting thin, and it's just more and more wear and tear that people think is the problem. But according to some new studies, it's not necessarily just wear and tear. There may have been injuries early on that triggered inflammation and a change in the metabolic profile of the joint so that it changes from a nice, happy joint with happy cartilage to a point where the chondrocytes, where there are cells within the bone, within the cartilage that start basically eating each other. They're like, we are in danger. We're going to destroy more bones. And these researchers at Penn State University were looking at bones and osteoarthritis and this condition in which the cartilage deteriorates. And they determined that there is a point at which a gene becomes involved called GSK2. And this gene encodes a protein that grabs onto a cellular receptor in the membrane of one of these chondrocytes. And in doing that, it overstimulates the cartilage destruction. So the GSK2 is very involved in destroying cartilage. And these researchers, they're like, oh, but hey, look at this antidepressant called paroxetine that is a GSK2 blocker. Let's just see what happens with the cells when we put this antidepressant on knee bones and knee cartilage. Whoever would have thought, you want to put an antidepressant on my arthritic knees? Make them happy? I don't know. It worked. They have seen that the antidepressant paroxetine is successful in blocking GSK2 in the cartilage, and in a mouse model of osteoarthritis, recovering cartilage so that the cartilage actually grows back.
This is like the
Which is
medical
ridiculous.
equivalent of finding a conch in a drawer in a museum, right?
Yes.
It's
Yes.
just like, we're using this thing for this, but maybe for this other thing.
Maybe for this, but it might work for this other thing. And that's the other point to this, is that paroxetine is already approved by the FDA for safety. And we know that it is effective in treating depression. So they won't have to do any drug safety testing. And they are in the process of applying to the FDA for clinical trial approval to test this antidepressant against osteoarthritis in people. They're going to move directly from mice to humans. They showed that it worked on human cartilage cells in a petri dish. We know that it works in a mouse, and they're like, hey, let's just take this to the clinical trial level because we know this drug is safe. So let's see if we can do this. The one thing that that it didn't say, that I couldn't find information about, was whether or not people who take paroxetine as an antidepressant have less problems with osteoarthritis as they age. And I think that would be a very interesting question to answer. I'd love to know that. Yeah. Yes. Timid tenor, Prozac kills cartilage. That is off-label on the knee prescribing. So it's very, very, it's pretty exciting. Pretty exciting news. And then my last story for the night is about depression. We've gone from Prozac for paroxetine for knees and cartilage to how your dad might be to blame for your adult depression.
I mean, in some cases, for sure.
What?
Yeah, so this is another mouse study published in the journal Science Advances. Researchers were looking at epigenetic changes in sperm and how they affect mental health. And in the study, they exposed male mice to a bunch of stress for 5 weeks, to the point where the mice started showing depressive symptoms. So depressed male mice, adults. And then they let them sire offspring. And they compared the offspring to a control group whose dads had never been stressed out. And they found that these mice that had depressed dads, they didn't show depressive symptoms normally, but then when they were exposed to a mild stressor, they got triggered. They became depressed. They showed depressive symptoms, increased immobility, decreased weight, and so the researchers in this study think that this appears to be linked to small RNAs in the sperm, and these molecules, they're regulating development and other aspects of health as the fetus is developing. Even through they are transmitted along with the sperm during fertilization, and that these small RNAs, as a result of it, increased corticosterone, cortisol levels in the fathers, are changing the kinds of small RNAs that are passed on at a particular time. And then those are related to anxiety disorders, depression in the offspring. So we'll see how this, whether this passes in the same way for humans. We don't know that this works in the same way, but it is very interesting to see this kind of a link in mice, and a social, cognitively advanced creature.
Yeah. So these aren't being reared by depressed dads. So this is a very interesting nature versus nurture sort of a study here, because obviously in humans it's going to be both.
Mm-hmm.
It's very likely both. Or you're going to absolutely be able to point to behaviors that you may have learned from or experienced from having a depressive or stressed parent, father. But separate from that, you're still going to be on the hook for it, even if you somehow manage not to be directly involved. That's really wild. I think thankfully my pops must not have had much stress. my pops, I'm gonna have to go ask, because he's actually a pretty chill guy, and kind of makes sense that even that we didn't have that close an upbringing, so there is that removal of nurture.
Totally. Yeah. You're an N of one, Justin.
So yeah, like I am. Like, in a way, I'm like, yeah, he's always been relaxed and chill in the face of pretty much anything. And I kind of have taken on that too. Interesting. Although
I mean, this is not completely causal. This isn't like, 100% your dad's stressed out. That's to blame. Your dad was stressed out.
Now it is. Now if you have no. Don't take that away from people, Kiki. Now they have somebody who they can blame.
Dad,
Absolutely.
Why were you so stressed out? Yeah. No, we don't. This is not 100%. We don't know all the details. This is a mouse study. It is very interesting, though, the passage of these epigenetic factors.
Yeah, with so much of these things that we've been learning about how these traits and things and reactions do get passed on through some bizarre genetic memory without mechanism. But yet, we seem to have, I think this is the first time I really recall, a mechanism involved in this isn't just a correlative. They're saying we know the mechanism by which this is transmitting.
Mm-hmm. Right. The mechanism by which
a big leap, but still awesome.
And there are going to be people who have genetic predispositions to anxiety and depression. There are potentially going to be people who have less of a genetic predisposition, but because of the environmental stressors that influenced dad, mom before you were made, a person is made, those environmental factors can influence your development or the offspring's development. And
yeah, but it does make sense that
how it's passed down. Yeah. Not just genetic, epigenetic, that stress in life can influence the next generation.
Yeah. I want to say there was a study that we reported on that involved grandmothers and something about food scarcity and it causing an epigenetic change in their grandchildren.
Yes, it skips a generation.
That was also fathers. Yeah. That was grandfathers.
I thought it was grandmothers.
As I recall, it was grandfathers. The one I'm thinking of, at least, is is it Dutch? farmers or
timid
Norwegian
tenor
farmers?
in the YouTube chat room is saying it sounds like the epigenetic studies of Norwegian families during
Norwegian.
famines.
Yes,
So it might have been the
yes.
Norwegians.
And that one was, well, it might have been both, actually, come to think of it. But it was, they had to be...
Everybody's starved at one point or another.
The grandparents, what determined the rates of diabetes and a bunch of different factors could get correlated back to prepubescent feast or famine times for grandparents. So it was, if they've experienced this as a 20-year-old, it had no effect on the following generation. But if it had before, I think boys at 12, girls at 13, 11, I don't know when things happen. But for both, it was, but I think the strongest hits that they had found, though, that were, when I remember, were the grandfathers. The grandfathers to the grandsons seem to have the strongest correlation. Anyway,
yeah. Kevin Unique is asking, how can they tell this? Can they prove the mothers are not depressed? I've never seen a mother not being depressed. Mothers are tired, that's for sure. But what they were looking at were very specific measures of what they would consider mouse depression, which are specific changes in behaviors that mice normally do. And they were specifically altering what was going on with the fathers, not with the mothers. So hopefully you would hope that they did pay attention to the mothers and whether or not the mothers were just displaying any behaviors or if they were just normal mice. But anyway, we have come to the end of the show. Thank you all for listening. Thank you. Time for shout outs. Shout outs to Fada for helping with show notes and with social media posts. I would also like to give a shout out to our new script supervisor showrunner, Rachel. Thank you for being here tonight in the chat room and keeping an eye on things. She's going to be assistanting and helping from here on out. So I'm very, very excited to welcome Rachel to the TWIS team. Thank you so much for joining us. Additionally, Gord, thank you for manning the chat room. Identity4, thank you for recording the show. And thank you very, very much to our Patreon sponsors for all of their generous support. Thank you to Woody MS, Andre Bassett, Chris Wozniak, Dave Bunn, Vegard Chefstad, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, John Scioli, Guillaume, John Lee, Ali Coffin, Gaurav Sharma, Shubru, Darwin Handon, Donald Mundus, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard Brendan Minish, Melazon, Johnny Gridley, Kevin Railsback, Flying Out, Kevin Porter, Christopher Dreyer, Mark Nazaros, Artyom, Greg Briggs, John Atwood, two fabulous thespians, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramos, Matt Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Sarah Chavez, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Stanton, Paul W. Stanton, Paul Disney, Patrick Pecoraro, Gary S., Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you for all of your support on Patreon. And if you are interested in having us read your name at the end of the show, head over to twist.org and click on that Patreon link. On next week's show, we will be speaking about Fungus February.
Yes.
With a fun guy, Dr. Ivan Liachko, and he will be joining us to talk about gene sequencing and funguses and other fun things. Don't miss it.
That's amazing.
Yeah. We'll also be back on Wednesday at 8 p.m. Yes, PM. That's what we do in Pacific time, broadcasting live from our YouTube, Facebook, and Twitch channels and twist.org/live.
Hey, do you want to listen to us as a podcast? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, please get your friends to subscribe as well.
Yeah, and for more information on anything that you've heard here today, show notes and links to stories are going to be available on our website, TWIS.org. You can also sign up for our newsletter.
Hey, do you want to contact us directly? Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, into the subject line, or your email will be spam filtered all the way into some knobbly knees, missing some cartilage, I guess.
Ah, ouch, achy. You don't want it to go there. You can also ping us on the Twitter. We are at Twist Science, at Dr. Kiki, at Jacksonfly, at Blair's Menagerie, and we really do love your feedback. There's a topic you want us to address, suggestion for an interview. Please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news. And if you've learned anything at all from the show tonight, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought. And I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.